Okay, so in this video, we'll be going over now part 2 of some IB questions, real past paper IB questions, paper 1. So specifically multiple choice, because that's paper 1. And it will be on the topic 3.5. So before you do this, make sure to check out part 1 and watch the videos where I explain everything that you need to know from 3.5. That will make these questions so much easier. Also, doing these past paper questions is the best thing you can possibly do, in my opinion, in the IB, because teachers in your class will tend to use the exact same questions from the past papers directly in the test, and so you're likely to see the exact same questions in your test when that you practice at home, and so you can get good scores automatically. So that's, in my opinion, the best thing you can do. So let's get to it. So I'll show it to you and you try it by yourself, and then I'll explain it as usual. So, which statement about the polymerase chain reaction, or PCR, is correct? So, before we look at the options, a real quick review on what PCR is. So, in a nutshell, PCR is the process that creates many copies of DNA, millions of copies of DNA, in a matter of hours from a tiny bit of DNA. So, for example, if you have a cell like this and you take out its DNA, and you put this single bit of DNA, this is from one single cell, into a PCR machine. So pretend this blue arrow right here is the PCR machine. This PCR machine will turn this single DNA, this single bit of DNA, into millions of copies within a matter of hours. This is useful because a lot of the testing machines that are used in crime scenes to determine who the murderer is or who the parent is in all that scenarios require a lot of DNA to work. They won't work off of a little bit of DNA. So that's why the PCR machine is extremely important. You can kind of think of it like a printer. If you put too little ink into a printer, it gives you a really blurry image, and so it's not so accurate. That's the same thing with this. You put too little DNA into this machine, it will give you an answer, but it won't be reliable. So a lot of DNA is important. So let's look at the options. So option A says PCR allows DNA to be cloned in a test tube and millions of copies can be made in hours. So that sounds correct. So let's see. Cloned, what does that mean to you? Cloned means to exactly duplicate. A PCR machine exactly duplicates this DNA. So all of these right here is going to be identical to this one. There will be no differences. It's exactly a clone. So far it's correct. It clones DNA in a test tube and millions of copies can be made in hours. Yeah, and that's exactly true. Millions of copies. They love this machine because it works really fast. So far, it seems like A is probably correct. But we need to make sure to eliminate the rest. So PCR allows the synthesis of RNA from DNA molecules. Oh no, this is definitely not true. We're not making any RNA molecules. You're only making more DNA from little DNA. So RNA is not even a single factor. So that's why RNA would make this question wrong. The process that does make RNA from DNA, if you recall, you would know transcription. That's when you make mRNA from DNA, but that doesn't involve anything to do with PCR. So this is incorrect. How about option C? So option C is saying PCR is a natural process carried out by some viruses to amplify DNA molecules. So, okay, this is incorrect because it's not a natural process. We build a machine to do it. Okay, that's why it's legit a machine that does it. It does it through a natural process because DNA replication is natural, but it's not natural by itself. We have to build it, and viruses don't do it. Viruses don't amplify these DNA molecules. So that's why this one is incorrect. Not a natural process. So next one, PCR is man-made technique. Yes, used to identify the nucleotide sequence of a DNA. So the first part is correct, second part is incorrect because by using this technique, you can't identify the sequence of the DNA. All you're doing is making more DNA. This technique doesn't zoom into the DNA to see what the sequence is. It just makes more. So this wouldn't be correct. So 15A, the answer for this one would be A. And the reason why it says 15, by the way, is because I take them randomly from past papers. And so this was probably question 15 in the past paper. So it's going to be random. Okay, next one. Let me open it for you. which technique separates proteins according to size? So the technique that does this, okay, I'll break down, we'll go through A, B, C, and D, and then eventually get to the answer, and I'll explain it in more detail. So A, treatment with restriction endonucleases. Okay, so this one is incorrect because restriction endonucleases, you can basically imagine a restriction endonuclease is like a scissor. What it does is it cuts DNA into little pieces. So when you treat, so these don't separate proteins according to size. All it does is it cuts up DNA. So that's what this thing does. So it wouldn't be correct for this case. And PCR, we just saw this one makes millions of copies of DNA from a tiny bit of DNA. So that doesn't do anything with size of proteins. Gel electrophoresis. So this process is correct because gel electrophoresis, I have a diagram here to help explain. So if you have, this is a gel electrophoresis machine, and you can see there's a positive side and a negative side, okay? Don't worry about the word electrode, just know this is the positive side and negative sides. And if you know, hopefully you know that positive things attract negative things. So if you put two magnets next to each other and they attract towards each other, that's because one is negatively charged and one is positively charged. Now, if you see these little slits here, these little slits here are called sample welds. And when you put proteins in there, so say we have Johnny Bravo here, and we put two random proteins in there, okay? So two random proteins. And we turn on the machine. What will happen is these proteins are negatively charged. So what happens is they are repelled by this negative charge, so they'll be pushed away by this negative charge towards this side. But at the same time, the positive charge here will pull it towards this side. So two things cause these proteins to shift towards this side. Interestingly though, this blue material here, called a gel, hence gel electrophoresis, this gel is like a glue, and it slows down these proteins from traveling fast. Because if there was no gel, these proteins would be here in a split second, and we wouldn't be able to tell the difference. So this gel slows it down, and the lighter protein will travel further because it's lighter. So you can imagine tying a rope around someone's waist that's light and a rope around someone's waist that's heavy. Obviously, when you pull them both, the lighter person will come towards you faster, right? So that's the same concept here. The lighter protein will move further in the same time that the heavier protein does. So the heavier protein moved a lot less in the same time. Therefore, a technique that separates proteins according to size is gel electrophoresis. Because after this experiment, you can tell which protein is heavier. The one that traveled further. The one that traveled least is heavier. Okay, why is the last one incorrect? So DNA profiling separates proteins according to size. Okay, so this is not correct because DNA profiling is the final image that you get. So for example, gel electrophoresis is the process that causes this image to be made, and this image is called a DNA profile. So this is not a technique. This is more like the final result. So a DNA profile is like the final result, whereas gel electrophoresis is the process that makes a DNA profile. Okay? Therefore, this one is incorrect because it's not really a process. It's more like a final result. Therefore, C is correct. 16C. Okay, let's go for one more. This next one is quite interesting. So, this diagram shows. I'll first zoom out a bit and give you the answers so you can give it a go yourself. Okay, so the diagram shows results of electrophoresis of DNA from a crime scene. So electrophoresis, we just mentioned, was when you put DNA in this kind of situation, and the charge will separate the DNA on the different people. So, for example, in this case, these lines right here are the same as these lines right there. So after the machine was turned on, the DNA traveled from these sample wells all the way down, depending on their size. And when you do this with different people, you'll get different results because different people have different DNA. And when their DNA is separated, it will separate differently from other people because their DNA is literally different. And so there's different sizes to each person, and so the final image will be different. Okay? So just bear in mind the concept that different individuals will have a different DNA profile, a different separation of the DNA fragments when you do an electrophoresis test. So let's look at this in more detail. What does this even mean? So we can see here the first two rows, so this one and this one, is the information from a crime scene. So when you walk into a crime scene, you normally see, you probably see two samples. Normally one of them would be from the victim, the person who was killed or injured or something, and the other one would be from the person who is the murderer, so the person who did the killing. Okay? So that's the purpose of these two samples. So one of these is from the murderer, and one of them is from the victim. So the question is, which one is from the victim and which one is from the murderer? So in this case, we could see that sample 2 here has the exact same separation of DNA as victim. So therefore, sample 2 of the crime scene is probably the victim. Now, what is the question actually asking? Which suspect could be implicated as a criminal according to the gel of DNA shown? So they're asking, which of these people right here, 1, 2, 3, or 4? So a suspect is obviously the person that is a potential murderer. It's someone that the police thinks could have done the killing. Now they're saying which suspect, 1, 2, 3, or 4, could be the criminal. So if we know sample 2 is the victim, and we already found the victim, sample 1 is the only thing left, which means this must be the sample from the murderer, then we know whichever suspect, 1, 2, 3, or 4, this sample matches is likely to be the murderer. So let's see. So I made the victim ones green because obviously they're innocent, so green is sign of good. So let's make this one red. Let's see if I can do this. Okay, so which one, 1, 2, 3, or 4, has the same separation of DNA as this sample 1? Okay, here. This one, right? You can see this suspect 4 is probably the murderer because their DNA shows the exact same separation as sample 1, which means that sample 1 and suspect 4 is the same DNA, which means suspect 4 was at the crime scene and was the murderer. So its answer will be D. Again, all of these questions, as you do them and you don't understand them, I really encourage you to check out the teaching videos I did because they explain all these things in detail. I don't have time to explain everything in detail here, otherwise there'd be no use in the teaching videos. So these are just to go over the questions that you already should have a good understanding of. So I'll make a part 3 again and do a couple of questions again, and part 4 and part 5 and so on, until I think I covered enough, and hopefully these are useful for you.